Welcome to our Central Dogma project by Johnny, Chase, and Connor. Starting off with DNA replication. DNA replication happens in the nucleus of eukaryotic cells, while in prokaryotic cells it happens in the cytoplasm because they don't have a nucleus. DNA nucleotides are represented by the colorful papers, where guanine is yellow, cytosine is pink, adenine is brown, and thymine is orange. The two carbon-nitrogen ring bases are adenine guanine, which are purines, while on the other hand, one carbon-nitrogen ring bases are thymine and cytosine, so those are our pyrimidines. To begin, helicase is an enzyme that splits the hydrogen bonds between base pairs on the original DNA strand, and basically they just unwind the DNA strand to create the leading and lagging strands. To make sure that DNA does not supercoil, topoisomerase, the pink hoop around the DNA strand, decatenates interlocked DNA and relieves stress on the strand. And moving on to the leading strand, DNA polymerase builds the new DNA strand starting from the RNA primer. And in other words, RNA primer initiates the process. RNA primers are synthesized by an enzyme called the primase, and RNA primers bind to the DNA strand in the 3' end of the parent strand so that the polymerase can build in a 5 to 3 direction. And keep in mind that RNA primer that binds to the original strand must also have binded with complementary base pairs in DNA strand. So basically that means that your base pairing is the same, except uracil replaces thymine in creating the RNA primer. Following this, DNA polymerase 3 will actually be the one that's building on the leading strand because it is the major replicative enzyme. Moving on to the lagging strand. Similarly, there must be an RNA primer to initiate the process on the lagging strand. However, since DNA must be synthesized in the 5 to 3 direction, we kind of run into a problem. Because the DNA strands are being unwinded by the helicase in the opposite direction, Okazaki fragments are thus created. And DNA polymerase 1 is the one that's constantly needing to go back every single time there's a new primer added. DNA polymerase 1 is present on the lagging strand because its function is actually to remove these primers and fill in these Okazaki fragments, where ligase, the last enzyme we'll be talking about, is the enzyme that specifically binds these fragments together. So here's an in-depth view of the nucleotide sequence, where since it's a DNA strand, we actually have a deoxyribose group on the 2' carbon, meaning that there's no oxygen in that position, in that 2' carbon position. The correct sequencing is, like I said above, the 5' to 3' direction, and phosphate groups are actually the ones connected to the 5' carbon, while the 3' carbon is attached to the hydroxyl group, shown by the OH. And lastly, types of DNA mutation. So first, we can have substitution, where you're changing the base sequence one for one, where you're changing a base for another base, but in doing so, you're actually changing the amino acid sequence, which can end up changing the protein's primary, secondary, tertiary, quaternary structures, and by doing so, you're actually changing the function of the protein as well. And you can change the function and the shape of a protein through insertion and deletion, which are actually frame shift mutations that can heavily impact the function of the amino acid, because you're basically shifting codons over.
Transcription first takes place in the nucleus for eukaryotic cells, and in prokaryotic cells, transcription takes place in the cytoplasm. Transcription is split apart into 3 sections: initiation, elongation, and termination. Starting with initiation, specific transcription factors assemble at a specific promoter along the DNA. The promoter acts as a region that allows the RNA strand to start being built. RNA polymerase is the builder that builds the strand. Other activator proteins help place the RNA polymerase into place. Contact between the activator proteins and the initiation complex releases the copying mechanism, where the RNA polymerase will begin to unzip a portion of the DNA helix, exposing bases on the strand. Since the RNA polymerase only attaches to one of the 2 DNA strands, we call the strand that it connects to the antisense strand, and the strand that it does not connect to is a coding strand. The antisense strand is a template strand for the mRNA. The length of the DNA that gallows this promoter is called the gene, and its assembly will eventually lead to the creation of a polypeptide chain, or protein. Remember that RNA polymerase is attached in the 5' to 3' direction. Next is elongation. During elongation, the RNA polymerase continues to build and attach nucleotides to a growing sequence. This continues until a stop codon is detected. Finally, when the RNA polymerase reaches a stop sequence, it will detach the RNA strand that it has built. Now we have our strand of either mRNA or a primary transcript. In eukaryotic cells, the result of this process is a primary transcript that needs to undergo processing before it can be considered an mRNA. During processing, there are 3 important steps: the addition of a GTP cap, the addition of a poly A tail, and RNA splicing. The addition of a GTP cap stabilizes the end of the microtubule to prevent polymerization and depolymerization. Poly A tail is added to make sure that the RNA molecule is more stable and prevents its degradation. It also allows the mature mRNA molecule to be exported from the nucleus and translated into a protein by ribosomes in the cytoplasm. RNA splicing removes the introns, which are the non-coding regions, from pre-mRNA to produce a final and functional set of instructions for the protein.
This is the process of translation. Translation occurs in the cytoplasm of both eukaryotic and prokaryotic cells. The process of translation is characterized into 3 different steps: initiation, elongation, and termination. Starting with initiation, a small ribosomal subunit is attached to the mRNA strand. At the same time, in initiation, tRNA molecule recognizes and binds to the mRNA molecule. Then, a larger ribosomal subunit joins this complex after it has been formed, and this complex will move along the mRNA until it reaches a start codon site on the mRNA. Ribosomes are needed because they read the sequence of nucleotides on the mRNA. They read these groups of nucleotides in groups of 3, which then results in the creation of codons. For every 3 bases, or 1 codon, 1 amino acid can be attached to a polypeptide chain. tRNA does the actual translating of the codon into an amino acid. One end is binded to the mRNA with corresponding bases in the anticodon. The anticodon is on the tRNA and the codon is on the mRNA. Moving on to translation elongation, amino acids are added to the polypeptide sequence. As ribosome complex moves along the mRNA strand, tRNA is released until the other tRNAs recognize the mRNA codon and take on the open position with its corresponding anticodon. By doing so, the amino acids that are attached to the tRNA attach to each other, forming polypeptide chains. Lastly, in termination, ribosomes reach a stop codon. Termination occurs when the mRNA codons do not have an anticodon on the tRNA. Release factors initiate termination.